Hello friends, today we are going to discuss the summary of the book Brain Energy written by Dr. Chris Palmer, the famous Harvard psychiatrist who offers an insightful look into the origins of mental disorders and the methods of treating them. Dr. Palmer's theory will usher in potent therapeutic interventions for pervasive psychiatric conditions for which the standard pharmaceutical approaches, that is, the psychiatric medications, have proven to be minimally effective. First, we will start by the fact that the psychiatric medications are not addressing the root cause of mental illness. Then we will be studying about the most important organelles of the cell, that is the mitochondria. Then we will study how the brain energy imbalance occurs and becomes the cause for various mental as well as metabolic disorders. Then we'll be studying about the different contributing causes which cause impairment in the mitochondrial function, and these causes include genetics and epigenetics. Epigenetics is the science which deals with the turning on and off of the genes. Then about the chemical imbalances, the neurotransmitters and medications which are contributing causes for mitochondrial impairment causing various types of disorders. Then about the hormones and the metabolic regulators. Next about the phenomenon of inflammation and how it is related to mitochondrial impairment. Then about the food, fasting and gut microbiome which is affecting our mental and metabolic health. Next about how physical activity affects our mental as well as metabolic health and how it impacts mitochondrial function. Also about the impact of sleep, light and circadian rhythms on our metabolism. And finally about designing a treatment plan for the various mental as well as metabolic disorders. Now we start with the fact that psychiatric medications are giving symptomatic treatments and they are not addressing the root cause of mental illness. We will explain it with the help of an example. We all have experienced common cold. We have suffered from common cold, and today we know that common cold is caused by the cold virus. But long back, when the cause of common cold was not known and researchers were still struggling to find out how common cold is caused and what are the methods of treating it, at that time they may have seen that common cold is causing symptoms. They may have seen that people are having symptoms like runny nose and sore throat. Now suppose person A is having runny nose, person B is having sore throat, and person C may be having both runny nose as well as sore throat. Now researchers might have seen that giving pseudoephedrine it treats the symptom of runny nose. So giving this drug pseudoephedrine, it treats the symptom of runny nose, and giving another drug Tylenol, it gives relief from the symptom of sore throat. Now they might have followed the trail further and tried to classify these groups, the people who are having runny nose like the person A, into the runny nose group and the people having sore throats into the sore throat group. And since giving pseudoephedrine corrected the symptom of runny nose, they might have tried to reason that these people with runny noses, they had a disorder called a pseudoephedrine deficiency disorder. And since giving Tylenol treated the symptom of sore throat, the symptom of sore throat went away after giving Tylenol. So the researchers might have tried to reason that based on the treatment, the people having sore throats are having a disorder known as the Tylenol deficiency disorder. But today we know that giving Tylenol is not going to cure common cold. Giving Tylenol is going to only give the relief from the symptom of sore throat. People may not have sore throat, but it will not cure the common cold itself. And regardless of whether a person is given Tylenol or not, either the immune system will actually fight the virus and the person will heal on his own, or the immune system may not be able to fight the virus and the person may die. So these two outcomes will occur regardless of whether Tylenol is being given or not, because Tylenol is only treating the symptoms. It is not addressing the root cause of the illness. And the path of progression of the illness will be the same regardless of whether Tylenol is given or not. And likewise for pseudoephedrine, regardless of whether this pseudoephedrine is given for runny nose or not, the illness is going to progress in the same way. Either the person will heal, that is the immune system will heal the person, or the person will die because the immune system is unable to fight the cold virus. So these symptomatic treatments are not addressing the root cause of the disorder or the illness, and they are merely addressing the symptoms. They are merely treating the symptoms, and hence the disease is not cured by giving these symptomatic treatments. Now we know that people with allergies, they have both runny nose as well as sore throat. And giving of antihistamines can actually cure both the symptoms of runny nose as well as sore throats. So antihistamines can cure both the symptoms, that is both the runny nose as well as sore throat. Now we see that the symptoms are occurring because of two different root causes. One root cause is the cold virus, today we know about it, and another root cause is the allergy. So people may have the symptoms of runny nose and sore throat because of the cold virus, or they may have the same symptoms because of a different root cause, that is allergy. Cold is caused by the cold virus and allergy is caused by different allergens. Now, two different root causes and same symptoms. Now, how do we explain this? The same symptoms are occurring because of two different root causes. So the author explains this by saying that there is a common pathway which is involved in both these disorders, in both these illnesses, that is common cold as well as allergy. Now, what is this common pathway? This common pathway is the pathway of inflammation. We know that both these disorders, that is the common cold as well as the allergy, they are caused through the common pathway of inflammation. The body goes through the process of inflammation and then these disorders occur. So this common pathway may explain same symptoms due to two different root causes. Now we come to mental disorders. What the mistake that we are doing in case of mental disorders is that we are only classifying the disorders based on the symptomatic treatments. Just as we classified the people with runny noses as having the pseudoephedrine deficiency disorder and the people with sore throats as having the Tylenol deficiency disorder, and it looked very irrational because today we know the root cause. We are doing the same mistake in case of mental disorders. And we are saying that depression is due to serotonin deficiency because giving serotonin is treating the symptoms of depression. The symptoms of depression are reducing by giving serotonin. And we are saying that psychosis is due to too much dopamine. So correcting the dopamine imbalance is treating the symptoms of psychosis. But what we are not seeing is that they are merely symptomatic treatments, and giving these treatments are not going to impact how the disease is going to progress. And the disease is not going to get cured by giving these symptomatic treatments. So this is the mistake that we are doing in case of mental illnesses according to the author. We look at the treatments that work and assume that it tells us the story of what is causing the disorders. So we just look at the treatments and we think that this is the story of what is causing the disorders. We are not addressing the root causes of mental illness. We are merely giving symptomatic treatments. And how do we classify these mental disorders? We are also wrongly classifying these mental disorders. What we are doing is that we are just looking at the cluster of symptoms. These disorders themselves, they are simply clusters of symptoms. We have seen certain cluster of symptoms and we are classifying it as a disorder. We are labeling it as a disorder. Now these diagnostic labels that we give, like depression, anxiety, bipolar disorder, schizophrenia, schizoaffective disorder, All these labels, epilepsy, all these labels that we are giving, they are meaning nothing in terms of the cause and effect. We are just looking at the symptoms, certain clusters of symptoms, and labeling it as a disorder. But we are not addressing the root causes, and we are not seeing. They just, these diagnostic labels, they mean nothing in terms of what is happening in the body and brain. So we are not digging deeper, we are just looking at the superficial things, that is the symptoms. Now what the author says is that these mental disorders, mental as well as the metabolic disorders, they are having a the mental disorders have a metabolic connection to them. So it is wrong to look at the mind and body in silos. It is wrong to look at mental and metabolic disorder in silos as separate entities, because they are interlinked, they are linked to each other. So both mental as well as metabolic disorders, they are linked to each other. Mental disorders have a metabolic connection to them, and the root cause of mental disorders might be a metabolic imbalance, a metabolic disorder. And also these mental as well as metabolic disorders, both they are occurring through a common pathway. And what is this common pathway? It is the mitochondria in our cells. Now then people may say that, okay, these mental disorders are occurring due to a common pathway of mitochondria, and mental disorders have a metabolic connection to them. But then why are there so much of diversity of symptoms of mental illnesses? So why are people who have the same disorders, the same diagnostic labels of mental disorders, why are they experiencing different symptoms? Some people with depression may have some symptoms. Like one person with depression may not talk to anyone and may not be interested in work. Another person with depression may have totally different symptoms. So why is this occurring? So the different symptoms of the people having the same diagnostic label in terms of mental disorders is due to certain pre-existing vulnerabilities. Certain pre-existing vulnerabilities are there. Because there are a number of factors which impact our cells, and a lot of environmental factors, and each person comes in contact with different environmental factors, and these pre-existing vulnerabilities can affect the intensity of the symptoms experienced by a person. So two persons having the same diagnostic label, there may be a difference in the symptoms experienced by them. There may be a difference in the intensity of the symptoms experienced by them. And the author says that there is a bidirectional relationship between the mental as well as metabolic disorders. Now, what is metabolism? Metabolism means burning of food to produce energy. So the process of burning of food to produce energy, that is the metabolism. Now, metabolic disorders include many disorders like diabetes, hypertension, high blood pressure. Hypertension is high blood pressure, or other physical disorders. And psychiatric disorders, we know that they have a connection with our mind. Then there are neurological disorders like epilepsy. So they have a connection with the neurons. Now we come to the most important organelle of our cell, that is the mitochondria. These are very primitive organelles because they were the organelles which were the first organelles formed, because earlier these mitochondria, they were not parts of the cell. Instead, they were an independent living organism. The first mitochondrion was a bacterium. So first mitochondria was a bacterium, and it had an independent existence. And researchers, they estimate that mitochondria evolved from an independent living organism sometime between 1 to 4 billion years ago. And a 1998 research paper suggests that they share many genes with modern day Rickettsia prowazaki, a bacterium that causes typhus. So it was earlier an independent living organism, a bacterium. Then another living organism, an archaea. It engulfed this ancestral mitochondria. So it ate up this mitochondria. But when it ate up this mitochondria, the mitochondria did not die. It continued to live, and the archaea also continued to live. It became the first eukaryotic cell. So both these, when they became one entity, they became the first eukaryotic cell. We know that eukaryotic cells are the mature or the developed cells, the primitive cells, they are called the prokaryotic cell. The prokaryotic cell do not have differentiated cell organelles. But the eukaryotic cell have a differentiated nucleus as well as cell organelles. The parts of the cell in a eukaryotic cell are well defined. So the first eukaryotic cell was formed when this archaea engulfed the mitochondria. And now what happened was that the mitochondria, it started performing the function of making energy. And this archaea, it started performing the function of fetching food for the cell. Now, the importance of mitochondria. Things that have persisted for a long time, they are thought to be more essential to life. Since the mitochondria are the oldest organelles in a way, so they influence the development of all other cell organelles. They influence the development of all other cell parts, and they became indispensable for the life of the cell. And hence other cell parts don't work correctly without mitochondria. Now this is a diagram of the mitochondria. Now we come to the modern mitochondria. Mitochondria are no longer able to replicate themselves outside of a eukaryotic cell. They no longer have an independent existence because in humans, most mitochondria transferred most of their DNA to the cell's nucleus. So they transferred most of their DNA to the nucleus of the cell, where the human DNA resides. We know that human DNA, it resides within the nucleus of the cell. So mitochondria, the mitochondrial organelle, it transferred most of its DNA to the cell's nucleus, where the human DNA resides. We know that DNA is encoding all the information that is related to an individual in the form of genes. Genes are segments of DNA, and these genes are recording all the information of the individual. Now, mitochondria transferred most of their DNA to the nucleus of the cell. There are now 1500, there are around 1500 mitochondrial genes which are now embedded within the human DNA. So these 1500 mitochondrial genes are embedded within the human DNA. And what do these genes do? They make proteins that are required to either create or maintain mitochondria. So these genes, they make proteins. So what do the genes do? They make proteins that are required to either create or maintain the mitochondria, and these proteins are shared with all the mitochondria in the cells. We know that genes are making proteins, and these proteins are performing large number of functions. So these mitochondrial proteins, these mitochondrial genes are making proteins that are required to create and maintain the mitochondria in the cells. Now mitochondria gave 1500 mitochondrial genes to the DNA in the nucleus, but they did not give all their genes. They still retain 37 genes with themselves. So 37 genes are still retained with the mitochondria. So they have some degree of independence. And individual mitochondria can use these 37 genes, these DNA on their own, and they maintain some kind of independence. Now, average human cell, it has around 300 to 400 mitochondria. And now each cell has 300 to 400 mitochondria. And the entire human body has got around 10 million mitochondria. And they make around 10% of our body weight. So mitochondria make up around 10% of the body weight of an individual. So we can see how important they are. And the brain cells, they require a lot of energy. So they are metabolically demanding cells. So in metabolically demanding cells, such as the brain cells, a single brain cell can contain thousands of mitochondria. Single brain cell can contain thousands of mitochondria, with mitochondria making up 40% of the cell volume of each brain cell. So in each brain cell, the mitochondria are making up 40% of the cell volume. So in the whole body, they are making 10% of our body weight, and in our brain, the mitochondria are making up, in each brain cell, they are making up 40% of the cell volume. So they are even more important for our brain. Mitochondria are even more important for our brain. Now we know that mitochondria are called the powerhouse of the cell, and they produce energy for the functioning of the cell. So how do they produce energy? They convert ADP, that is adenosine diphosphate, into ATP, that is adenosine triphosphate, by adding a phosphate group. So this is the process of making energy, that is ATP molecules. So small amount of ATP can also be produced by a process known as glycolysis without mitochondria. But the lion's share of ATP, the majority of ATP, it is the lion's share, that is the major share of ATP, especially for the brain cells, is produced by the mitochondria. In average human adult, mitochondria make about 9 multiplied by 10 to the power 20 molecules, 20 ATP molecules every second. So they are very hardworking organelles. And one group of researchers looked at brain cells using specialized imaging techniques and found that a single neuron in the human brain uses about 4.7 billion ATP molecules every second. So a single neuron in our brain uses 4.7 billion ATP molecules every second. Now we know that the neuron is having a cell body called the cytone. This is the cell body. Then there are dendrites which are protruding from the cytone. Then there is the axon. The axon is covered by myelin sheath which is protecting the axon. Then there are the nerve endings, and this is connected to the dendrites of another neuron. The dendrites of another neuron. This is another neuron. And then this is next neuron. And what is this gap? This small gap between these two neurons, it is known as, this small gap, it is called as the synapse. It is called as the synapse. Now we'll study that mitochondria are found at these synapses also, because these are the places where the things are actually happening. Synapses play a very important role in nerve impulse transmission. One group of researchers looked at brain cells using specialized imaging techniques and found that a single neuron in the human brain, it uses 4.7 billion ATP molecules every second. So single neuron requires so much of energy. We can see how important mitochondria are for the neurons in our brain. Now we come to mitochondria, a property of mitochondria, that is they move around living cells. They are moving around, but not all mitochondria are moving around. Some mitochondria, they are not moving and they appear to stay in places where things are always happening. Now what are these places where things are always happening? That is near the factories where proteins are made. So that is the ribosomes. So ribosomes are an important organelle in our cells. They are macromolecular machines found within cells that perform the biological protein synthesis. So what do ribosomes do? They make proteins. So these mitochondria, they are found near ribosomes and also near synapses, because synapses they are the gaps between the nerve cells and they play an important role in nerve impulse transmission. So here is where the mitochondria that are not moving around, they are found. Now we know that energy needs to be produced in the right amount, at the right place, at the right time, and it goes through a very fast recycling process which is involving mitochondria. And mitochondria, they are rapid recyclers. ATP, we know that it is the energy currency of human cells, and when ATP is used as energy, a phosphate group is removed and it turns into ADP. Now what happens is mitochondria are turning ADP into ATP by adding a phosphate group. Now when this ATP is used as energy, then this ATP gets reconverted into ADP and releases a phosphate group. So mitochondria, then what will happen is the process again starts. Mitochondria are taking this ADP, again converting into ATP by adding a phosphate group. So this goes on. And mitochondria take ADP, turn it back into ATP by attaching a phosphate group, and then transfer it to the cell cytoplasm. So they transfer the ATP to the cell cytoplasm, that is the liquid that is filling the cell. The cytoplasm is the liquid between the cell nucleus and the cell, and the nuclear membrane and the plasma membrane of the cell, that is the cell cytoplasm. So in that cytoplasm, the mitochondria are transferring this ATP because it is needed in the cytoplasm. Majority of the cell organelles are there in the cytoplasm. They give one ATP and recycle one ADP simultaneously. So this process of recycling of ADP and producing of ATP, all of this is going on simultaneously. if there is not a mitochondria or two in the right place, if mitochondria are not there at the right place at the right time to deliver all the ATP or recycle all the ADP, then things will either slow down or stop working. So mitochondria are the workforce in the cells, and they need to be at the right place at the right time. If they are not there at the right place at the right time to deliver all the ATP, that is the energy, or to recycle all the ADP into ATP, then things will either slow down or they will stop working. So as we will study further, these mental and metabolic disorders, they are occurring because of the mitochondrial dysfunction, because mitochondria may not be performing their function properly, they may not be at the right place at the right time, and because of them that our cell activity may slow down or the cells may stop working, and this may lead to disorders. Now, mitochondrial movement is more important than just making sure enough energy is supplied at the right time at the right place. So, mitochondrial movement is very important. And if this mitochondrial movement is disrupted, then it may cause mental as well as metabolic disorders. As we are going to study further, there is a protein called tau, which is impairing mitochondrial movement. And this protein tau, it accumulates due to maintenance problem in the cell, due to debris accumulation in the cell, and this tau protein is impairing the mitochondrial movement, and it becomes a cause for Alzheimer's disease, which is a neurodegenerative disorder. So mitochondrial movement is very important, and if it does not take place, then disorders occur. It is all related to mitochondrial interaction with other organelles and with each other. So mitochondria, they are interacting with other organelles by moving around. Mitochondria are the only organelles in the cell which move around. All other organelles are not moving around, they are stationary. So mitochondria are going to the other organelles, they are interacting with other organelles, and they are facilitating the function of other organelles. And if this doesn't happen, mitochondria are interacting with other organelles as well as with each other. And if this is not happening, then disorders occur. These interactions are critically important to almost all cell functions and to gene expression. Now, what is gene expression? Gene expression means the gene performing its function. So, the activating of the gene. So, gene expression means turning of the gene on. When the gene is turned on, it starts performing its function. So, mitochondria are very important for gene expression. This is the science of gene expression is also known as epigenetics. Now, the neurons, they have a resting membrane potential. Now, what is this resting membrane potential? So, this is a cell, this is the nucleus. Inside of the cell has got a negative charge as compared to the outside of the cell. So, inside of the cell is having a negative charge. And this charge, this negative charge, is critically important for the functioning of the cell. And how does this negative charge occur? This negative charge, it is created by ion pumps, which pump sodium, potassium, calcium and other ions either inside or outside the cell. So there are certain ion pumps which are pumping these sodium, potassium, calcium and other ions either inside the cell or outside the cell. These pumps are pumping these ions, or they are pumping these ions between compartments within the cell, or they are pumping these ions between compartments within the cell. So these ion pumps, they are creating this negative charge. And for these ion pumps to work, energy is needed. And where is the energy provided from? This energy is provided from the mitochondria. And cells are doing a lot of this ion pumping in order to be ready to fire. So in order to be ready to fire, that is to start functioning, the cells are doing a lot of ion pumping. And when these ion pumps, they are creating a negative charge inside the cell as compared to outside the cell. And this is known as resting membrane potential. And when the cell is triggered, that is the cell starts functioning, it becomes ready to fire. It sets off a cascade of events. So when the cell starts performing its function, which may include releasing a hormone or releasing a neurotransmitter, it sets off a cascade of events that results in the cell doing its tasks. So cell will do its task, whether it is releasing a hormone or releasing a neurotransmitter or doing something else. So different cells may have different tasks. Now to stop the cell from doing its task, like stopping the release of a neurotransmitter or stopping the release of a hormone. How do you stop the cell from doing its task? It requires energy. Now the cell is doing its task, so it can't keep doing its task forever, because suppose the body requires a certain hormone. When the deficiency is corrected, when the body has sufficient amount of that hormone, the cell will need to stop working. Because if the cell still keeps on working, then it will cause another imbalance, because more hormone is not required. So the cell needs to stop working. So how does the cell stop working? So when a car stops working, we apply brakes. To make a car stop, we need to apply brakes. So to make a cell stop, we need to apply brakes. Applying brakes in a car requires energy. Similarly, applying brakes in the cell requires energy. And this energy, it is provided by mitochondria. So to stop a cell, when a cell is working, and to stop it from working, to stop its work, we need to provide energy, and that is provided by mitochondria. When the work of the cell is no longer needed, it needs to stop, and that energy is provided by mitochondria. Now mitochondria are involved in calcium regulation. And when mitochondria are prevented from functioning properly, calcium regulation is disrupted. Now, when mitochondria are not functioning properly, then this regulation of calcium in the cell, it is disrupted. And the off switch, that is turning the cell off, the switch off for the cell, that is the brake to the cell, that can also be disrupted, because calcium plays a crucial role in switching the cell off. And when this calcium regulation is disrupted due to mitochondrial dysfunction, then the off switch of the cell may get disrupted, and the cell may become hyperexcitable. The cell may become hyperexcitable. So mitochondria are playing an essential role in the off switch in the cell. Now mitochondria provide the energy needed for ion pumping. Mitochondria, they provide the energy needed for ion pumping and also regulate the calcium levels in the cell. And these calcium levels are functioning as essential off switches for the cells. When the cells are energy deprived, then what will happen? Then the brakes on the cells are not being applied. The cell is functioning and the brake is not being applied, because brake needs energy. So some cells will stay on for too long, and while other cells, they will fail to work. So this hyperexcitability of cells can occur if the cells are energy deprived. And this energy deprivation may occur because of mitochondrial dysfunction. Now, psychosis. Psychosis, which is a symptom of schizophrenia and schizoaffective disorder. Now, this is a symbol of hyperexcitable cells, because we know that when we are sleeping, then certain cells, when they become active, we have dreams during sleep. And this is because of certain cells that become active while we are sleeping. Now when these cells which are producing dreamlike experiences, they need to shut down when we wake up. And when these are not getting shut down, because there is no energy for applying brakes, then we might have these dreamlike experiences even while we are awake. And these dreamlike experiences, in the language of psychiatry, they are called as hallucinations. That is, people start to see things, or they start to hear things that are actually not there. This is because of hyperexcitability of certain cells. So psychosis. So this hyperexcitability occurs because of energy deprivation in the cells when there is mitochondrial dysfunction. So addressing mitochondrial dysfunction can help in curing of psychosis. This is what is being suggested by the author. Now we will just see the role that is played by mitochondria. Mitochondria help regulate the metabolism of the cell, of or body broadly. So mitochondria play a role in regulating of our metabolism. They help produce and regulate the neurotransmitters. So they are helping in the production of certain neurotransmitters. They play a direct role in the production of certain neurotransmitters like estrogen. Sorry, these are hormones. In production of certain neurotransmitters, they are playing a direct role like serotonin or dopamine or glutamate or norepinephrine. Then they are also regulating the release and functioning of neurotransmitters. Mitochondria help regulate the immune system function. They are regulating how our immune system is functioning. They regulate our stress response. Mitochondria is regulating our stress response. How we respond to stress. Mitochondria, they create reactive oxygen species and they help clean it up. Now, reactive oxygen species, they often cause damage to our cells. They may play a beneficial role also, but excess of reactive oxygen species may start damaging our cells. And mitochondria, they help clean it up. They reduce their toxicity. Then mitochondria are playing a role in gene expression, that is turning of the genes on. So gene expression, that is epigenetics, turning genes on and off. Mitochondria play a crucial role in this. Then mitochondria can multiply, they can fuse with each other, they can divide, multiply, they can form long tubular chains. Mitochondria are also involved in cell growth and differentiation. So in cell growth and differentiation, mitochondria play a crucial role. Mitochondria play a crucial role in neuroplasticity. Neuroplasticity means forming of new neural networks through cell growth and differentiation in the brain. So mitochondria play a crucial role in that. Then they also help maintain the existing cells. Maintenance function is an important function of mitochondria. They eliminate the old and damaged cells. So they eliminate the old damaged cells and help in forming new, they help in recycling of the cell parts, forming of new cells. Now this is a diagram of the animal cell. We can see in the center is a nucleus. Then we have various organelles that are surrounding the nucleus, and here is the mitochondria. Then the other organelles, they include the endoplasmic reticulum, then the ribosome, which have an important role in producing of proteins in protein synthesis. Then we have the lysosome. Lysosomes are known as the suicide, and they help in recycling of waste materials. They are known as the suicide bags of the cell. They help in eliminating the old and damaged cells. And mitochondria play a crucial role in the regulation of the in the eliminating of old and damaged cells, and they facilitate the functioning of lysosomes. Then we have the Golgi apparatus, the vacuole, and then we have a cell membrane that is covering the animal cell. Now we come to the functions of mitochondria. We have just seen the functions, we will study them in greater detail. Now mitochondria are helping in regulating the metabolism broadly. Now these are certain peptides, humanin, MOTSC, SHLPI6. These are certain peptides, and the genes for these peptides are on the mitochondrial DNA. And these peptides, they are produced by the mitochondria. So genes for these peptides, they are on the mitochondrial DNA, and these peptides, they are produced by the mitochondria. Now what is a peptide? Peptide is a molecule made of 2 or more amino acids. So we know that amino acid is the basic unit for making of proteins. Now when 2 or more amino acids, they are linked, then they make a peptide, and most genes, they contain information to make functional molecules. We know that genes, they have information for making of functional molecules, and these functional molecules are the proteins. And peptides are in a way constituents of these proteins. Peptides are made when 2 or more amino acid molecules, they are linked to each other. So when two or more amino acids, they are linked to make a molecule, they make peptides. Now the genes for the peptides are in a way proteins. They are a constituent unit of the proteins. Genes for these peptides are on the mitochondrial DNA. We know that genes are coding for proteins. So genes are coding for these peptides or proteins. And the genes that are coding for all these peptides, they are on the mitochondrial DNA. And these peptides, they are produced by mitochondria. And what is the beneficial effect of these peptides? These peptides, they have beneficial effects on illnesses like Alzheimer's, strokes, diabetes, heart attacks, and certain types of cancer. They also have broad effects on metabolism, on cell survival, and on inflammation. Now, the existence of these peptides suggests that mitochondria are able to communicate with each other through these peptides. So these peptides, they help the mitochondria to communicate with each other. These peptide signals help the mitochondria to communicate with each other in order to regulate the metabolism throughout the body. So these peptides are helping the mitochondria to communicate with each other for regulating metabolism. And these peptides, they have beneficial effects in illnesses like Alzheimer's, strokes, diabetes, heart attack, and certain types of cancer. So this is about how mitochondria communicate with each other using the peptides, and they regulate our metabolism. Then the next function of mitochondria is mitochondria produce and regulate the neurotransmitters. So they are playing a role in production of neurotransmitters and also in the regulation of neurotransmitters. Now mitochondria, they play a direct role in the production of acetylcholine, glutamate, norepinephrine, that is a neurotransmitter that helps us focus. This norepinephrine deficiency is found in people with ADHD. Then dopamine. Dopamine is playing a role in psychosis. It is said that dopamine occurs due to too much of dopamine is a symptom of psychosis. Too much of dopamine occurs in case of psychosis. So psychosis is a symptom of too much of dopamine. Then GABA. GABA is also a very important neurotransmitter. Now GABA plays a role in inhibiting or stopping of hyperexcitable cells. So GABA is playing a role in inhibiting or stopping of hyperexcitable cells. Then serotonin. Serotonin is playing a role in depression. So it is said that deficiency of serotonin may cause depression and anxiety disorders. Now, how do the mitochondria help produce and regulate neurotransmitters? Now neurons, the nerve cells, that is the neurons, they have one specific neurotransmitter that they specialize in making. So each neuron may be specializing in making one neurotransmitter. Now production of these neurotransmitters, energy is required for the entire process of production of neurotransmitters, in the regulation of neurotransmitters. So energy is required. Energy is produced by mitochondria. It is required in the production of neurotransmitters. Then the vesicles, they travel down the axon to the release site. So the vesicles that are the cell organelles, the vesicles, they store these neurotransmitters. First, the neurotransmitters, they are produced. They are produced and mitochondria are playing a role in their production. Then once these are produced, they are stored in the vesicles. Neurotransmitters, they are stored in the vesicles. And these vesicles, they travel down the axon of the neuron. They travel down the axon to get to their ultimate release site. Now this is a diagram of the neuron. Now neurotransmitters are stored in these vesicles. Then this is the neuron. This is the cell body of the neuron. Then this is the axon. This is the nerve ending. This is the myelin sheath which is covering the axon. Now, neurotransmitters, they are stored in these vesicles. This is the vesicle. These vesicles, they travel down the axon and they travel to the release site. Now, this is another nerve cell body of another nerve cell, dendrites of another nerve cell, another axon. Now, this is the synapse. This is the synapse, that is the gap between two nerve cells. Now, the vesicles are traveling down the axon to the release site. So, the release site may be a synapse. And here the neurotransmitter may be released. It is produced, then it travels down the axon and it is released. And this also takes energy. Release of neurotransmitter. Traveling of this vesicle needs energy. Release of neurotransmitter requires energy. And once the neurotransmitters, they are released at one location, then the mitochondria, they will move to another location. Since their work is over, they will move to another location of the cell membrane to release a new batch of neurotransmitters. So wherever in the cell, the energy is required, wherever in the cell the neurotransmitter is required, the mitochondria are going to move to that location. In this way, the neurotransmitters are released. And once the neurotransmitters have their impact on the target tissue, now they are released and then they will have an impact on the target tissue. Then the target tissue or the muscle or the gland cell. So once the neurotransmitters have their impact on the target tissue, on the target muscle or the target gland cell, and after they are released from the receptors of the target cell, now the neurotransmitters will go to the target cell. Now target cell has got certain receptors and they will be received by the target cell. Now when their work is done, when the neurotransmitters are given to the cell, then they will be sucked back. They will be sucked back into the axon terminals. They will be sucked back. This is called as reuptake. Now see, first the production of neurotransmitters, mitochondria are involved in this. Then they are stored in the vesicles and the vesicles travel down the axon to the release site. Now the signal to release neurotransmitters, it depends on the resting membrane potential and the calcium levels. So the signal, where does the signal to release neurotransmitter comes from? It comes from the resting membrane potential and the calcium levels in the cell. So that is the negative charge in the cell, the resting membrane potential and the calcium levels in the cell, they affect the signal to release the neurotransmitter. Once the neurotransmitter is released on the target cell, then they are sucked back. First they are released, and when sufficient amount is released, they will be sucked back into the axon terminals. Now this is the nerve cell, this is the axon, and they will be sucked back. The neurotransmitter is going to be sucked back into the axon terminals, and this is known as reuptake. And this reuptake also requires energy. And finally, they will be repackaged into the vesicles. Again, they will be repackaged into the vesicles for the next round, for going to the next target tissue. So this is how the production and release of neurotransmitters, it is regulated by mitochondria. Now, mitochondria also have receptors for some neurotransmitters. So cells, as we have seen that the target tissue, it has receptors for neurotransmitters. Mitochondria also have receptors for some neurotransmitters. So some neurotransmitters, mitochondria are playing a role in the release of neurotransmitters, in the production of neurotransmitters. Now these neurotransmitters are also affecting the mitochondria. So there is a feedback cycle. The feedback cycle is that the mitochondria are playing a role in the release of neurotransmitters, in the production of neurotransmitters, and the neurotransmitters are also affecting the mitochondria, because the mitochondria also have receptors for these neurotransmitters. These mitochondria also have some enzymes that are involved in the breakdown of neurotransmitters. So these mitochondria, they also have certain enzymes which are involved in the breakdown of neurotransmitters. These enzymes include monoamine oxidase enzyme. And what does this enzyme do? This enzyme it is helpful in the breakdown of certain neurotransmitters like dopamine, like serotonin. We'll be studying about this. Now, they are involved in regulating the release of GABA. Now, mitochondria are involved in the release of GABA, which is a neurotransmitter. And we know that GABA neurotransmitter, it plays a role in stopping it plays an inhibitory function. It acts as an inhibitory neurotransmitter. So what it does is that it slows down the hyperexcitable cells. It slows down the hyperexcitable cells, and when this GABA is not getting released, then these cells will not be slowed down. These cells will become hyperexcitable, and this may result in symptoms like psychosis or anxiety symptoms. So mitochondria are regulating the release of this GABA neurotransmitter, and mitochondria actually store GABA within themselves. Then several neurotransmitters are known to regulate mitochondrial function, production and growth. So we know that there is a feedback cycle. The mitochondria are playing a role in the production and release of neurotransmitters, and neurotransmitters are also impacting the mitochondria. They are regulating the mitochondrial function, production and growth. So there is a feedback cycle. Now the next function of mitochondria. The next function is that mitochondria, they help regulate the functioning of the immune system. Now mitochondria help regulate how the immune cells engage with the immune receptors. So mitochondria are helping regulate how the immune cells are engaging with the immune receptors. When cells are highly stressed, then they often release components of mitochondria. So when the cells are stressed, it leads to inflammation, and they often release certain damage associated molecular patterns, DAMPs. And mitochondria are some of these damage associated molecular patterns. Some of them also include mitochondria. So when cells are stressed, they often release components of mitochondria in the form of damage associated molecular patterns, and these serve as a danger signal to the rest of the body, and this activates chronic low-grade inflammation. So this is how mitochondria help regulate the immune system function. So mitochondria are playing a role in inflammation. They help regulate how the immune cells engage with the immune receptors. One study looked at specific types of immune cells called macrophages to see how these cells coordinate with the complicated repair processes in wound healing. So how these macrophages, that are a type of immune cells, how they coordinated the repair processes in wound healing. Now these macrophages, they do different things during different phases of wound healing. So macrophages are doing different things during the different phases of wound healing. Until this study, it wasn't known how the cells know when and how to change between phases. So wound healing is involving different phases, and the macrophages are required to change from one phase to another. Earlier it was not known how the cells know when to change between phases, but researchers found that mitochondria specifically controlled these processes of how macrophages play a role in wound healing and how they change between the different phases of wound healing. So mitochondria are in a way helping the macrophages to coordinate the repair processes in wound healing. So this is how mitochondria are regulating the immune system. Then mitochondria are also regulating our stress response. Now stressors can be of two types. One is physical stressors and another is mental stressors. Now physical stressors can include starvation, infection, lack of oxygen, and mental stressors is anything that is threatening or challenging us. It may have a very big psychological impact on us. Anything which threatens us or challenges us. Now, integrated stress response through changes in metabolism, gene expression, and other adaptations. So, mitochondria are involved in this integrated stress response. So, how the stress response takes place? The body will either change its metabolism or the gene expression will be changed. in response to stress, or other adaptations may take place. And many lines of research show that mitochondrial stress itself leads to integrated stress response. So many lines of research say that stress will directly impact mitochondria. It is said that stress can increase production of reactive oxygen species, and these reactive oxygen species can damage mitochondria, and it can cause mitochondrial stress. So mitochondria are helping regulate our stress response. How are they helping in regulating our stress response? By changes in metabolism, by changes in gene expression, turning genes on or off in response to stress, and other adaptation. Now, if a cell is itself unable to manage stress, if a cell is not able to manage stress on its own, then two things can happen. One is that the cell will trigger its own death. So it will commit suicide. It will trigger its own death, and this is known as apoptosis. So apoptosis means the cell is triggering its own death, or it enters into a zombie-like state, or the cell will become kind of inactive. It will enter into a zombie-like state, and this is known as senescence. And this cell entering into a zombie-like state is associated with aging and many health-related problems like cancer. So this is what happens when a cell is unable to manage stress. Either it will commit suicide, that is apoptosis, or it will enter a zombie-like state, which is also called senescence. And this cell entering into zombie-like state is associated with aging and many health-related problems like cancer. Now mitochondria, they play an important role in coordinating different aspects of psychological stress response in the body and brain. Then mitochondria, they are involved in making, releasing and responding to hormones. So mitochondria play important role in responding to hormones, in releasing hormones and making hormones. Hormones like estrogen or thyroid, cortisol. So mitochondria are playing a direct role in the production of these hormones. For this, energy is needed, ATP is needed, and this energy it is delivered by mitochondria. So production of hormones requires energy, and this energy is given by mitochondria. Mitochondria in other cells sometimes have receptors for these hormones. Now, mitochondria are regulating the release of hormones, and hormones are also affecting mitochondria, because mitochondria in other cells may have receptors for these hormones. So there is a feedback cycle. Now, mitochondria create reactive oxygen species, and they also help clean it up. Mitochondria create reactive oxygen species and help clean it up. So mitochondria, our bodies, they burn fuel. Now either carbohydrate, fat or protein, we burn fuel to produce energy. And now burning fuel will sometimes also create waste products. There will be waste products. And when mitochondria are burning the fuel, then electrons, they flow along the electron transport chain. And these electrons, they are the source of energy used to make either ATP or heat. These electrons are the source of energy which is required for making ATP or heat. And most of these electrons are within the usual system. They are flowing along the electron transport chain. But sometimes these electrons, they leak outside the usual system. Sometimes they leak outside the usual system. And when they get leaked, when they leak outside the usual system, then they form reactive oxygen species. And now reactive oxygen species, they are molecules like the superoxide ion, that is O2 minus, or the hydroxyl radical, that is OH minus, or organic peroxides, H2O2. So these are examples of reactive oxygen species. Now most of us, we treat these reactive oxygen species as toxic, and we treat them as toxic waste products, but they are not simply toxic waste products. Small amount of these reactive oxygen species also serve as useful signaling process inside the cell. So some of these reactive oxygen species, small amount of them, they also serve a useful signaling process. Useful signaling process inside the cell. So they are also useful to the cell in some way. Now, a study says that reactive oxygen species, they are primarily regulators of heat production and energy expenditure. So they also help in regulating the heat production and energy expenditure, which is a broad measure of metabolic rate. So they regulate heat production and energy expenditure, which is a broad measure of metabolic rate. So we can see that these reactive oxygen species, they are not only we can't say that they are only toxic. They are also having a useful role. They serve as useful signaling process inside the cell. They also regulate heat production and energy expenditure, which is a measure of metabolic rate. But if these reactive oxygen species, if they are in large amount, if they become uncontrolled, if they are in large amount, then they become toxic, and they may result in inflammation and oxidative stress. So reactive oxygen species, when they become uncontrolled, when their number increases too much, then they become toxic. Then they may result in inflammation and oxidative stress. Now, when these reactive oxygen species, they are in excess, they are known to cause damage to mitochondria as well as to the cells, and they are associated with aging and many diseases. Since these reactive oxygen species, they are produced in mitochondria. These reactive oxygen species, they are produced in mitochondria. We have already seen that when these electrons burning fuel will create waste products, and when mitochondria burn fuel, the electrons are flowing along the electron transport chain, and when some of these electrons leak outside, they form reactive oxygen species. So mitochondria are playing a role in the production of reactive oxygen species, and they are produced in mitochondria, and they are highly reactive. They often damage mitochondria first. So when they become uncontrolled, they often damage mitochondria first. Why? Because mitochondrial DNA, it is unprotected. The nuclear DNA is protected, it is protected by the nuclear membrane, or it is properly protected. But mitochondrial DNA, it is vulnerable because it is unprotected, and hence it is more vulnerable to the impact of reactive oxygen species. So large amount of reactive oxygen species, they cause mutations in the mitochondrial DNA, and this can damage the mitochondrial machinery itself. So they are causing mutations in the mitochondrial DNA, and it can damage the mitochondrial machinery itself. And if they leak outside the mitochondria, they can damage different parts of the cell. So they can damage the mitochondria. If they leak outside, they can damage different parts of the cell. Now we have just seen about reactive oxygen species. Now mitochondria are also serving as the caretakers of the reactive oxygen species. They are serving as janitors or caretakers for reactive oxygen species. They produce these reactive oxygen species, and they also help clean up these reactive oxygen species. They help clean up some of the reactive oxygen species through an elaborate system of enzymes and other factors that serve to detoxify these reactive oxygen species. Through enzymes and other factors, the mitochondria are helping to detoxify the reactive oxygen species. And when this detoxification system, it fails, then reactive oxygen species waste products, they can pile up. Then the ROS will pile up, and they will cause damage, and this can lead to cellular dysfunction, otherwise known as aging, cell death, disease. So we have just seen that mitochondria are helping in producing reactive oxygen species as well as cleaning up reactive oxygen species. Then mitochondria change shape in response to different environmental factors. So depending on different environmental factors, the mitochondria, they change shape. Environmental factors means factors from outside. Now this changing of shape of mitochondria in response to environmental factors, it is very important to the functioning of the cell. They also interact with each other in profound ways. So mitochondria, they interact with each other in profound ways through fusion. They fuse with each other, or fission, or dividing. Research says that mitochondrial fusion impacts fat storage, eating behavior, and obesity. So storage of fat, eating behavior, obesity, it is impacted by mitochondrial fusion. Changes in the shape of mitochondria and fusion of mitochondria appear to create signals. So when mitochondria, they are fusing with each other or they are changing their shape, then it appears to create signals that can affect the entire body. And when the mitochondria, when they are prevented from doing these things, when the mitochondria, they are prevented from doing these things, then metabolic problems occur. Not just in the cells affected, but also sometimes in the whole body. So mitochondria are changing shape in response to different environmental factors. As we are going to study later on, intermittent fasting. When we are fasting, then mitochondria are repairing themselves. Damaged cell parts are getting destroyed and new ones are getting made. Some cell parts are getting recycled, and the mitochondria, the damaged ones are getting recycled, are getting destroyed and recycled, but some of the mitochondria, they change shape and they fuse with each other, form long tubular chains. And these mitochondria, which are fusing with each other, forming long tubular chains, they are providing the energy that are required for recycling of the cell parts, for producing new cells, for producing of new mitochondria. So some damaged mitochondria are getting replaced, while other mitochondria, they are fusing with each other, forming long tubular chains, and they are providing energy needed for the process of mitophagy, that is repairing of mitochondria, and autophagy, that is repairing of the cell. Now mitochondria are also playing a role in gene expression, that is turning genes on and off. Human genome, that is the genetic information of humans. Human genome, that is all the genes in the humans. They carry all our genetic information and they reside in the nuclear DNA. But now we know that it is not about the genes themselves, but about what causes these genes to turn on and off. So it is not the genes themselves, but it is the turning on or off of our genes that is impacting our metabolism. And this turning of genes on and off is known as epigenetics. And mitochondria, they are primary regulators of epigenetics. They regulate which genes are getting turned on and which genes are getting turned off. How do they regulate epigenetics? They send signals. Mitochondria, they send signals to the nuclear DNA in several different ways. So mitochondria are sending signals to the nuclear DNA, and in this way the genes within that DNA, they are either getting turned on or they are getting turned off. So when the gene is turning on, then the cell starts performing, then the gene starts making protein, performing its intended function. And when it is getting turned off, then it stops its function. So mitochondria are important regulators of epigenetics because they send signals to the nuclear DNA in several different ways and regulate the turning on and off of the genes. Now, gene expression. What is gene expression? It is the process by which the information that is encoded in the gene, it is turned into a function. So genes have got information related to the organism that is encoded in them. Suppose a gene in the heart will have information relating to how the heart is going to function. Now the genes will remain dormant till the time it is not turned on. So when this gene is turned on, then it starts functioning. Then that gene will start making the protein which will regulate the functioning of that particular organ. So the process by which information encoded in the gene is turned into a function, it is known as gene expression. And how is gene expression affected? What are the factors that are affecting this gene expression? So these factors include ratio of ATP to ADP, and the levels of the reactive oxygen species, the calcium levels in the cell, and all these are impacting gene expression. And all of these, they have direct relation to mitochondria. They have direct relation to mitochondrial function. Now mitochondria are required for transport of an important epigenetic factor. That is a factor which is regulating gene expression, which is turning on or off, which is involved in turning on or off of the genes. And what is this factor? Nuclear protein histone H1. So mitochondria are required for transporting this nuclear protein histone, which is regulating gene expression. So this protein histone, it is helping regulate gene expression, and mitochondria are involved in transport of this nuclear protein histone. And this nuclear protein histone, it is transported from the cytoplasm to the nucleus. It is transported from the cytoplasm. Now this is the cell, this is the nucleus, and the liquid that is filled in between the cell membrane, the plasma membrane of the cell and the nuclear membrane, it is known as cytoplasm. So this protein histone, it is transported from the cytoplasm to the nucleus, and this protein is regulating gene expression. And this process of transport, it requires energy, and this energy is provided by the mitochondria. And mitochondria must be present for the transfer of this protein histone. Now in 2030, it was discovered that mitochondrial reactive oxygen species directly inactivate an enzyme called histone dimethylase, which regulates epigenetic gene expression in cell nucleus. So mitochondrial reactive oxygen species, they inactivate this enzyme which regulates gene expression in the cell nucleus. And this process was found to play a role in extending the lifespan in yeast, and thought to possibly play a role in humans as well. So this process was playing a role in extending the lifespan of yeast. So we can see that mitochondria are involved in gene expression. Mitochondria are involved in the transport of this protein histone, which is regulating gene expression. Now next is that mitochondria, they can multiply. They can fuse with each other, they can multiply. They are involved in cell growth and differentiation. Now, we often hear of a term called neuroplasticity. What is neuroplasticity? It is the ability of the neural networks in our brain. So this is our brain. There are different neural networks in the brain. So the ability of the neural networks in the brain to change through growth and reorganization. So these neural networks, their ability to change through growth and reorganization is known as neuroplasticity. And this neuroplasticity is very important for various functions of the brain. When we grow, then our brain develops by means of this process of neuroplasticity. Our brain forms new neural connections. When we learn new information, then new neural connections are formed in our brain. When we learn new skills, new neural connections are formed. And when we grow up, our brains mature, they develop through the process of neuroplasticity. So the ability of these neural networks to change through growth and reorganization is known as neuroplasticity. Now, what is cerebral cortex? Cerebral cortex is the outermost layer of the brain, which is made up of billions of nerve cells that form the brain's grey matter. So cerebral cortex, it is the outermost layer of our brain. So the outermost layer of our brain, which is made of billions of nerve cells and which we call the grey matter. This is the cerebral cortex, and it is responsible for many higher brain functions, including memory, thinking, learning, etc. Learning, reasoning, problem solving, emotions, consciousness, sensory functions. Now our cerebral cortex, it has got four lobes. The frontal lobe, which is involved in the higher mental activities like cognition, memory, etc. Then the parietal lobe, the temporal lobe, and the occipital lobe. The occipital lobe is involved in vision. Temporal lobe is involved in regulating the activity of hearing. Parietal lobe regulates our sense of smell and other functions. So these are the four lobes in our brain. Now the neuroplasticity process, the process of growth and differentiation, it involves activation of specific genes in the right cells at the right time. So how do these neural networks change through growth? So it requires activation of specific genes. When genes get activated, they produce certain proteins. And this is how the growth and reorganization of neural networks takes place. So it requires activation of specific genes. As we have already seen, what is activation of genes? It is epigenetics. Turning off genes on and off, and mitochondria regulate epigenetics. So mitochondria are playing a direct role in activation of specific genes which are playing a role in neuroplasticity in the brain. Also involves many signaling pathways. So growth and differentiation of neurons also involves a lot of signaling pathways. And it involves production of building blocks for new cells and new cell parts. Involves production of building blocks. So building blocks need to be produced for new cells, new cell parts, growth and reorganization. Balanced with energy needs. So mitochondria are essential for producing of building blocks for new cells, new cell parts, and for because production of new cells, it requires energy, and energy is provided by mitochondria. So mitochondria are essential for the process of neuroplasticity and regulation of calcium levels and other signaling pathways. So we know, we have already studied that mitochondria are involved in regulation of calcium levels in the cell, and which is regulation of calcium levels is very essential for turning the cells off. So mitochondria are directly involved in this. Mitochondria are also involved in other signaling pathways. Then fusion. Mitochondria, we know they fuse with each other. Fusion of mitochondria, it appears to send signals that activate genes in the nucleus. So when the mitochondria, they are fusing with each other, then it appears to send signals that activate certain genes in the nucleus. And by activating of these genes, certain functions are performed which aids in neuroplasticity. So mitochondria, we can see they are sending signals. Fusion of mitochondria is sending signals which is activating genes in the nucleus. Then when mitochondria are prevented from doing this, when they are prevented from fusion or from moving around, then the cells will not develop correctly. Obviously, if the mitochondria are prevented from doing their task, then the cell will not develop correctly because the signals will not be sent, so genes will not be activated. So if genes are not activated, then cells will not grow and differentiate, and the process of neuroplasticity for the brain, that is growth and reorganization of brain cells, brain neurons, will not take place. So we can see how mitochondria are directly related to brain function, how they are crucial for brain function, how proper health of mitochondria is extremely crucial for the functioning of the brain. So when mitochondria are prevented from doing their tasks, cells will not develop correctly. Mitochondrial growth and maturation is essential for cell differentiation. So we know about several neurodegenerative disorders like Alzheimer's, like autism. So in that case, mitochondrial dysfunction may be playing a role in this, because when the mitochondria are not doing their task, when they are not performing their function properly, then activating of genes will not take place. Genes are not activating, then neuroplasticity is not taking place. New neural connections are not being formed, and this may lead to brain dysfunction. So direct and essential role of mitochondria is there in the development of brain cells. Now mitochondria help maintain the existing cells, and they also eliminate the old and damaged cells. So mitochondria, they help in maintaining the existing cells and also eliminating the old and the damaged cells. Now there are two terms. One is necrosis, and another is apoptosis. Now necrosis means sudden death of the cell. When the cell is abruptly killed, then it is known as necrosis. For example, when the cells die due to heart attack, the cells they die abruptly. So that is necrosis. Then apoptosis instead is a programmed cell death. Apoptosis means programmed cell death. Now apoptosis plays a useful role, and mitochondria are directly involved in programmed cell death. Now when the cell gets stressed, when the cell gets old, when the cell becomes old, when it becomes damaged, then what will happen? it will initiate its own death. So there is a planned process by which programmed cell death takes place, and because the signal to die comes from the cell itself. Because if the old and damaged cell stays too long, then it will adversely affect the other cells. So it needs to die, and the signal to die comes from the cell itself. And mitochondria are controlling this programmed cell death, because the older cells, they need to die for recycling of cell parts for new cells to be formed. And mitochondria are directly playing a role in this programmed cell death, which is called as apoptosis. Then now how does this apoptosis take place? When reactive oxygen species increase, when stress increases, then the cells, the mitochondria, they release. When mitochondria are experiencing high levels of stress and when they accumulate large amount of reactive oxygen species, then they begin to degrade. And when this happens, when these mitochondria, they begin to degrade, then the mitochondria, they release a protein called as cytochrome C. And this protein, cytochrome C, it activates killing enzymes called as caspases. And these caspases, they kill the cell. They lead to death of the cell and recycling of some cell parts. Some cell parts will get recycled and form new cell parts. So this process is very essential for regulating our metabolism, because old cells, damaged cells need to die, and new cells need to take their place in order to ensure that our metabolism is functioning properly. Now, what happens in case of stress response when a cell is stressed? It includes changes in 4 domains. One is the hypothalamus pituitary adrenal axis. We know these are glands. Hypothalamus gland is actually regulating the pituitary gland, and which is regulating all other glands. So stress response includes changes in 4 domains. One is the hypothalamus pituitary adrenal axis, which results in cortisol flowing through the bloodstream. So changes in the hypothalamus pituitary adrenal axis, it leads to cortisol. Cortisol is the stress hormone. It results in the stress hormone flowing through the bloodstream. The second change is the change to the sympathetic adrenal medullary axis, which results in adrenaline flowing through the bloodstream. Then the next change is inflammation. Inflammation may occur as a result of stress. Now, adrenaline gland is having 2 hormones. One is epinephrine, another is norepinephrine. Norepinephrine hormone is also helping us focus. And a deficiency of this norepinephrine hormone can lead to ADHD also. Now, another stress response includes another change, which is changes in gene expression. Because when the mitochondria are stressed, then it may impair mitochondrial function, and since mitochondria play a direct role in gene expression, so stress can cause changes. I will explain it again. Stress response includes changes in 4 domains. One is cortisol flowing through the bloodstream, another is adrenaline flowing through the bloodstream, next is inflammation, and third is changes in gene expression. So in response to stress, mitochondria, they send signals, or they themselves act as signals, and they play a role in changing of gene expression, that is controlling which genes are turning on and which are turning off, especially in the hippocampus. So hippocampus is an important part of our brain, which is involved in the consolidation of our memory. It plays a role in converting our short-term memory into long-term memory, and it plays a role in many other important functions of the brain as well. So changes in gene expression, especially in the hippocampus, take place in response to stress. Then adverse childhood experiences. So certain children might have experienced certain abuse in their childhood. So these adverse childhood experiences, they also increase the risk of mental disorders later in the life of children. Now, what happens when there are high levels of stress? It will inhibit autophagy. So what is autophagy? Autophagy means the cell's process of removing the old and damaged parts and repairing itself. So the cell repairing itself is autophagy. When the cell removes its old and damaged parts, it is known as autophagy, and autophagy is essential for metabolic health. So if stress increases, then this autophagy it is inhibited. Then when the cells are stressed, they slow the process of making new proteins. So when the cells become stressed, then they slow down the process of making new proteins. So this may act as detrimental to metabolic health. Sequestering messenger RNA. Now, when the cells are stressed, they slow the process of making new proteins, sequestering messenger RNA molecules into little bubbles called stress granules. Now, these messenger RNA molecules, they are directly involved in the making of proteins. So, these messenger RNA molecules, they are directly involved in making proteins. And when the cells are stressed, they slow the process of making new proteins, sequestering these messenger RNA molecules into little bubbles called as stress granules. So these messenger RNA molecules, if the cell was not stressed, then they would be involved in the function of making proteins, which would facilitate metabolic health, which would facilitate our body functioning properly. But when the cells are stressed, then these molecules, they are sequestered into certain stress granules, and they are not performing their function. New proteins are not getting made, and when new proteins are not getting made, then it may become the cause of neurodegenerative disorders. And high levels of cortisol can be caused. High levels of cortisol is always in response to stress, and there is a lot of stress, then a lot of the stress hormone is produced, and so high levels of cortisol stimulate the production of these stress granules, which lead to impairment of function of these messenger RNAs, and these messenger RNA do not make new proteins, and this can cause neurodegenerative disorders. Then stress can cause sleep disruptions, we all know, leading to higher cortisol levels. So it can cause sleep disruption, and stress can cause premature aging. So we will again summarize all this. What happens due to high levels of stress? One is autophagy, that is the cell recycling old and damaged cell parts and repairing itself. This process is inhibited. Then when cells are stressed, then the process of making new protein, it is slowed down. Then when cells are stressed, sleep disruption occurs. Now when the process of making new proteins is slowed down, then neurodegenerative disorders can occur. Also, stress can cause premature aging. Now this was about the most important organelles of the cell, that is the mitochondria, and all the different roles that the mitochondria are playing in our body in maintaining of our metabolic health. Now, why are different mental and metabolic disorders caused? They are caused because there is a brain energy imbalance. And because of this brain energy imbalance, this brain energy imbalance, it is caused due to mitochondrial dysfunction or mitochondrial dysregulation. So either the mitochondria are not functioning properly due to certain intrinsic factors, or they are not functioning properly because of certain environmental factors or extrinsic factors. So when the mitochondria are not functioning properly, then there is a brain energy imbalance caused. And this brain energy imbalance can lead to either hyperactive cells, overactive cells, or underactive cells, or absent when the cells are not functioning, absent cell function, or developmental disorders, neurodegenerative disorders, etc. So mitochondrial dysfunction can directly cause these hyperactive cells, underactive cells, absent cell functions, or developmental or neurodegenerative disorders. Now hyperactive cell functions can be seen in cases of psychosis, in case of heart arrhythmia, irregular heartbeat, in cases of heart arrhythmia, then psychosis, or in certain anxiety, certain cells becoming hyperactive can cause anxiety also. Then ADHD. No, ADHD is not due to hyperactive cell functions. Psychosis is a good example. Then underactive cell functions. So when the cell is slowing down, cell has slowed down due to lack of energy. Now hyperactivity can also be caused due to lack of energy because the brakes are not being applied on the cells. Underactivity can also be caused due to lack of energy, slowing down of the functioning of the cell. Then absent cell functions. The cell stops performing its task due to lack of energy. And neurodegenerative disorders, they can also be caused due to mitochondrial dysfunction. So this was about the mitochondria and how their dysfunction leads to various disorders. Now in the later part of the slides, we will be studying what is brain energy imbalance, how mitochondria are resulting in brain energy imbalance, about the various overactive cell functions, the underactive cell functions, the absent cell functions, etc. So in this part we have covered about mitochondria. This is part 1 of the video. In the next part of the video, we will be studying about brain energy imbalance. Now with this, we come to the end of this video, and the next part of the video, you must listen to the next part of the video to further understand the process of how the brain energy is regulated and how energy imbalance leads to various mental and metabolic disorders, and how Dr. Chris Palmer offers an insightful look about how to cause the root cause of mental disorders, and which involves the metabolic connection in all mental disorders. With this, we come to the end of this first part of the video, and please listen to the next part of the video for gaining further insights into the work of Dr. Palmer regarding the treatment of various mental and metabolic disorders. Thank you.